So my name is Stanley Lee. I'm an assistant professor in the clinical research division at Hutchinson Cancer Research Center in Seattle. I started my lab very recently, roughly about 18 months ago. Prior to that, I did my postdoctoral research at Memorial Sloan Kettering Cancer Center in New York. And before that, I actually traveled halfway across the world. So I did my graduate studies in Melbourne, Australia. And I did my undergraduate degree at the University of Auckland in New Zealand. And prior to that, we're an immigrant family, and I was originally born in Taiwan. So we've kind of come right around the world to, eventually ending up in Seattle. Yeah, so our laboratory is very interested in studying the process of blood cell formation, also known as hematopoiesis. This is a very dynamic process that happens, starting from the bone marrow of your body, where the hematopoietic stem cells, the cell that's responsible for producing all the different blood cell types in your body, work to do this process. And obviously this is a very important process because it's very important to have all the different blood cell types working in your body at any given time to sustain life and all the necessary things that you need to do to go about your everyday life. And I'm particularly interested in studying this because this process is actually very much regulated by a lot of different so-called molecular mechanisms. And when this process goes wrong, this is when you have really serious diseases, including cancers such as leukemia. So my lab primarily wants to understand at the molecular level how this process is governed, how they are regulated, and what happens when this process go wrong, and why this process go wrong in the context of serious life-threatening diseases such as bone marrow cancers, leukemias, and anemias. And most importantly, we want to use this knowledge that we gain from this our study to really develop better therapeutics for cancer patients, and this is really our primary goal of our research program. So I got really interested in science, I think starting from a very young age. When I was a kid, I remember reading from storybooks, to kids around the house, on astronomy, the star, and Copernicus and Newton, and later on I came across a story about Edward Jenner, who had studied the cowmaid. To learn about how having been exposed to the cowpox might have made her more immune to smallpox. And that was a really fascinating story. I think I remember I was like maybe 5 or 6, or just before elementary school, and that that sort of made me really become fascinated by biology in general. And later on, as I went through the schooling system, I had a lot of emphasis or interest in science, and especially biology. And I think during high school, that's when I really became interested and thinking that this could be a viable option for me as I enter university. So and that's exactly what I did. So when I went to university, I was enrolled in biomedical science degree at the University of Auckland. And from there on, I became very interested actually initially in neuroscience because I had a very awesome neuroscience professor in my first year, and he really inspired us to become interested in that. From that point onwards, I became very interested in stem cells and that sort of era. And I think that's the time around the time when the human genome was published. And after that, I decided to pursue graduate studies, and that's when I moved to the Walter Eliza Hall Institute in Australia to study a blood stem cell formation, and that kind of carried on through my postdoc, and here I am today. So I officially started here at the Hutch in October 2019. Yeah.
Right before
COVID. Yeah, right before COVID. Yeah.
Wow.
Yeah, so quite often when we're doing research, especially at the bench, we will come across a lot of uncertainties because a lot of times things just don't work. And the reason for that is primarily every single experiment, even though conceptually they could be extremely simple, it actually involves a multitude of steps that goes into it. And every step has to be exactly done correctly or close to perfect for you to see the result that you anticipate, and most importantly, results that you can also reproduce multiple times, after the first experiment. And that is for everybody a steep learning curve. Everyone picks up slightly, at different pace, different techniques. Some people are more are better at doing one technique over the other, and so therefore, when one first starts out in the lab, a lot of time we spend is on troubleshooting and perfecting our techniques. Scientific results are often based on the little things you do in the lab, and that will, require a lot of, practice. So, and then once you go after that, then you start asking, meeting some certain biological questions that may or may not work out the way you had predicted or hypothesized. And that's totally okay, because that means you have to explore other alternatives, and that's the fun part about science. So I think as you progress through your training, or one progresses through their training, I think you become more resilient because of that. And then failed experiments or things that don't work become very, I guess, a very common part of your life, and you learn to deal with it. And I think that builds resilience. And for me, that's what keeps me motivated and wanting to work harder to get that answer that I'm hoping to get.
My name is Tayyib. I'm a medical student, originally from Morocco. I'm here at the Hutch doing some cancer research, and my current title is a graduate research assistant. On a daily basis, what I do is a lot of cell culture or tissue culture, which I'll show you in a bit. And after that, I do a bunch of molecular biology experiments, which involve checking protein expressions in different cells, checking gene expressions in different cells, and another part is checking growth of different cells that we subject to different conditions. I'm in my last year of medical school. What I have left is a few clinical rotations, and after that, residency. So I'm planning on continuing in internal medicine because I'm hoping to join hematology oncology fellowship eventually, hence my current research in cancer. During the third year of medical school, we usually do clinical rotations, or what we call clerkships, and those are through the different services, so OBGYN, pediatrics, internal medicine, and so on and so forth. I opted to go for this research elective, which is basically an extra year added to your medical school. And so I chose that because I'm really interested in hematology oncology research, and I thought this is a great opportunity for me to gain some cutting-edge research in oncology and gain some more skills in this field, because hopefully I'm planning on becoming a clinician scientist, and this will definitely add on to my career. Well, I grew up in Morocco until the age of 18. I wouldn't say that during school I was sure of what I wanted. I always felt like there's always a pressure in the Moroccan system to sort of decide very early on, but there isn't really like it's not always clear to you as a student. You don't really get to experience a lot of things or a lot of careers. Fortunately for me, I was exposed to the medical field through my father, who was a physician at the time. But I wouldn't say that there's a clear event in my life or in my schooling that sort of made me decide that that's what I want to do. I feel like it's a combination of things, and then a bunch of hardships. I was raised by a single father who eventually got me where I am today, and just that compassion, that empathy, that sacrifice that I saw in my father growing up. And eventually I realized that patients are just people that you really have to care about, not only physically and also emotionally. I got to experience that as a rotating student in my clinical rotations. And just that human connection to me is really important. And that sort of constellation of things made me decide that this field or this profession is really what I want to continue doing for my life. I'm a musician. I've been playing piano for about 11 years now. I started in Morocco. I studied music for about 6 years. And then once I started undergrad, I had to sort of stop on going to classes for music and so on. And so science took over at that point. So I'm classically trained. However, I also got interested in jazz and a little bit of blues. I wouldn't say I'm great at it. I'm still learning. I also got into composing music, so I also make some of my own music at this point. And yeah, so that's pretty much what I do in my spare time, which I have plenty of. Today I'm going to be showing you tissue culture or cell culture. So broadly speaking, when scientists are doing experiments, they need model systems. These model systems are basically what allow us to do different experiments and sort of observe what's happening to these model organisms, allow us to uncover what certain proteins or certain genes are doing to the organism or to the system as a whole. So today I'm going to be showing you some leukemia cells that I've been culturing. What I did to these leukemia cells, I've introduced a gene in them, and what I'm trying to see is how does this gene affect their growth. Now, when you introduce a gene inside a cell, not all of the cells that are around will have taken that gene. It's just an imperfection in the experiment. However, we have this really cool trick that allows us to differentiate the cells that have taken this gene in as opposed to the ones that haven't. And this trick is basically making the cells that have taken this gene in look green. So tagging them with a green color. As opposed to the other ones which will not have that green color tagged to them. So first of all, I'm going to show you the cells. So this is our incubator where we keep our cells. So we keep them happy. Okay. So these are our cells. So you're not able to see the green color here because we need to put them under a different kind of light for you to be able to see that. To separate them from each other, we use this other technique called flow cytometry. And it's a technique that basically uses lasers that they shine at the cells, and the cells based on the laser, they're able to detect the green color, and it's able to separate the green ones from the non-green ones, and that way we're able to collect them in different tubes. So basically a purification technique. Okay, so what I'm going to do is basically prepare these cells for the flow cytometry or the purification. In our bone marrow, there are these stem cells that are responsible for giving rise to different cells in our blood. So the red blood cells, the white blood cells, and the platelets. We are trying to see, because this mutation is very frequently found in leukemias, we're trying to see if this mutation is causing the cells to remain stuck in an early form or in an immature form. After separating the cells that do have the gene of interest from the cells that don't, we will start from an equal number of cells and do a growth curve. And what that means is tracking the growth of these cells over time and seeing whether their growth is repressed or promoted. Are they growing faster or are they growing slower, as opposed to the control, which are the cells that do not have the gene that we're interested in. So the second experiment that we're interested in is a differentiation assay. Basically what this assay entails is checking if having this gene of interest is causing these cells to remain stuck in an early stage or an immature stage. And what we do is basically have these cells, after they have been purified, put them in a similar plate like this one. This is a 6-well plate, and add a cytokine, erythropoietin. The cytokine drives the cells into making, or the stem cells into making erythrocytes, which are the red blood cells. And so what we're interested in seeing, once we have this gene inside the cells and we add erythropoietin, are these cells able to become erythrocytes or progenitors to those erythrocytes, or are they stuck in an early form or an immature form and therefore not able to become mature red blood cells eventually. The way we do that is by checking for different markers at the surface of the cells that allow us to differentiate between mature red blood cells from immature red blood cells. What I mean by marker is a protein found on the surface of the cells, and by using an antibody that has a specific color tagged to it, the antibody will bind to the protein at the surface or on the surface of the cell, and flow cytometry allows us to detect that color and say, okay, this cell has this color, therefore this cell has that surface or that protein on its surface, or that surface marker, and therefore it's a red blood cell, an immature red blood cell. And basically combining that with the other experiment, which deals with the cell's ability to grow, whether fast or slow, we can basically develop a phenotype and basically say that, well, this gene is causing the cells to grow faster, but it's also causing the cells to not differentiate or remain stuck in an immature form. And so you may end up with cells that are growing very fast but are immature. And that's basically the idea behind leukemia. And I think this is something that I struggled with in high school. The fact that you're very uncertain about where you want to go, what profession, what career, and yet you feel like you're under pressure because you have to decide now. Point is, it's okay to be uncertain, because that and if you accept it, that way you're able to sort of look at what your options are. What your options are, explore. So explore different fields, and that's okay. If you end up in a field that you don't enjoy as much, you can always turn back and go to a different field. And I feel like in high school, that's really something that you should come to terms with and be okay with the uncertainty. There's always those sort of inflection points where you are sort of deciding, should I go there, should I go there, should I go there? I'm not sure, I don't know. But I think it's always very important to be uncertain. It's always important to recognize that you're uncertain, and based on that sort of explore your different options, especially at a very young age when you're still in high school. It's completely okay to be uncertain and explore different career choices.
So my name is Erica Arriaga Gomez, and I am a research technician too. And what I do is I provide support for like various lab things. So I place the orders, I make sure that things are stocked up, and then I also do some experiments. So I help out with some of the projects that people are working on, and so in particular the PI Stan, if he has things that he needs to do, and he maybe doesn't have time, he sometimes will like let me do them, and so then I just report back to him the results and let him know. I've been here a year, since January 1st. So yes, right before the pandemic. So I went to a small liberal arts school in Minnesota, and then I worked there as a research technician for a year. And then after that, I came to Fred Hutch.
How did you go from Minnesota to Seattle?
Because Seattle's it has way better weather, so that's how.
Better weather?
Oh yeah, definitely. Yeah, so I was actually really interested in coming to work at the Hutch just because of the mission statement, and then also based on a lot of the employee reviews I had read, it seemed like a lot of the employees really enjoyed the work environment. They said that it was very cohesive, very cooperative, and just people who were very enthusiastic about science, so that seemed great to me. In high school, I went to a science and math school, and so just as a kid, I always was very curious about like why do things happen, and my dad in particular was very encouraging about me asking questions like that. And so then growing up, I just really liked science and math, and so then when I was in high school, I was in an environment where that was encouraged. And one of the things that I did was, in high school, I volunteered to help out at a cancer center in Oklahoma. And that was when I think I really got to see kind of like the impact of cancer and how it really does affect people's lives. Like it doesn't just impact the person who has cancer, it also impacts their family around them. And so just during that time, I got to see how much people, like, they really needed support, and how it really was like it took like almost an entire village to help people through that time. In college, I had the opportunity to do a summer research program after my first year of college, and that really got me interested in questions about how do cells communicate, what are things that make, like, why do cells do the things they do. And so that, just going back to what I had seen in high school, it really motivated me to kind of just think of like, how can we make people's lives better by answering some of these questions, right? And so how can treatments get better? How can they be more efficient and less grueling and taxing on people, so that they can have a better quality of life? And so, yeah, so it was just scientific curiosity, and then also seeing, I guess, the impact of some of the scientific research. So for me, I do want to go to graduate school and do like immunology. And so right now I'm just working as a research technician, so I can gain more experience and just continue working in the scientific field. One thing I think I learned in college is that there's not a direct path, right? There's not one path to getting to your goals, and I think that just right now, I really like the idea of like learning new ththings. I'm trying to learn how to play the guitar. Emphasis on the trying part. It's going. I think it's just something that I always wanted to learn, and I think with the pandemic, like, again, it's not something that I'm doing. I don't have the consistency, I think, yet, but I just want to like encourage myself to learn something outside of the lab. I think for me personally, again, being a perfectionist and being a little type A, I guess, I kind of like expect things to go a certain way, and sometimes I'll be like set and like, this is how things are, and like I think just learning to cope with that, with just like, sometimes things don't work out, or sometimes, you know, life is messy and it's not, things don't, you know, there's not a one path to from like point A to point B. And I think really understanding that and embracing that, you know, like I look back at like when I was 18 and I was like, no, like, you know, I have to go to college and then I have to go to like grad school, you know, and like just kind of like being very like, I have to do this, and this. But then now I think I'm more flexible about things. I just kind of have realized that life is very messy and there are unforeseen circumstances, or like sometimes things, it's the timing isn't right for things, and I think it's always better to just, you know, if your goal is to go to professional school, to be a doctor or to be in grad school, not focus too much on like, I have to get there, more focus on like ensuring that you are, that, you know, like you pay attention to the journey of getting there, right? So like making sure that you are, you know, preparing yourself for it, or that you are giving yourself the time that you need to be in grad school or be in med school, like things like that. So I think, yes, just understanding that sometimes you have to take various different pit stops, and that's perfectly okay. I think, yes. You're really young, so yes.
My name is Elizabeth Bonner. My official job title is Graduate Student Research Assistant. It's a glorified graduate student. And kind of what my day-to-day looks like, it really actually widely varies depending on which day. So I take classes, so I may be in class. There's portions of my day, of my year where I've had to TA, so I'm teaching class. There are days where I just stay home and read articles. Yesterday I spent the day making slides for final presentations and lab meeting. And then my favorite days are the days that I get to sit here at the bench and do science and hopefully make interesting discoveries that change the way that we approach disease. My official affiliation is University of Washington School of Medicine MCB program. And then within the MCB program, we actually have access to, I think, 3 or 4 different research institutes. So that's how I wound up here at Fred Hutchinson Cancer Research Center. So this is my second year of graduate school. We're in the quarter system, so I'm entering my last quarter. And then next year, upon the start of my 3rd year, I'll be taking my general exam and I will officially move to the title of PhD candidate. So that's very exciting. So for your first year of graduate school, you do what's called rotations, and the length of those rotations varies depending on your institution, but here they're 10 weeks long. And you rotate through 3 different labs to see, do you like the science? Do you like the people? Is the mentorship style of your ultimate thesis mentor kind of align with you? And then after those 3 rotations, you then join the lab, and that will be where you do the entirety of your thesis work. So I started my undergraduate at Community College of Denver. It was a small little community college, and I got all of my prereqs out of the way there. So they had guaranteed transfer courses. It was way cheaper to start off at a small community college. And then I transferred to University of Denver, which is a small private institution. So I finished off my degree there with a degree in a BS in molecular biology and a concentration in neuroscience, and then I double minored in mathematics and chemistry. So I'm a non-traditional student. I actually didn't complete my high school education in high school, and I wound up just bartending and waitressing for many, many, many years. And some things went wrong in a bar that I was working at that made my safety feel jeopardized. So I decided I'd go back to school for nursing. I liked being on my feet, I liked, rushing around and feeling challenged, and I liked dealing with people. And so I was taking these, prerequisites at Community College of Denver, and I went to Denver's first charter school. So it was very experimental. It was one of the first charter schools in the nation. And I didn't have math and science. So none. So it wasn't even on my radar in high school. And I was sitting in my first cell biology class and I was like, wait a minute, what? I didn't even know what was going on. And I was so stoked on it. It was so cool. And I just knew that's where I want to be, because I would ask questions and my teachers would be like, well, we don't know, and they don't know, and we don't know. And I was just like, oh yes, this is absolutely where it's at. So I scrapped nursing. My dad thought I was nuts. He's, because it's a long path, and, as a student, you don't always make a lot of money. He thought I was absolutely crazy. And I was like, no, Dad, you don't get it. This is what I want to do. And of course now he's changed his mind and is fully in support and sees how much joy it brings to my life. But yeah, I was just blown away by what goes on inside the cell and how intricate and beautiful of a ballet it is. And I just couldn't imagine my life without that on a regular basis.
And you ended up minoring in math, right? Was
Yeah.
that one of your minors?
Yeah.
That's crazy.
And once again, I sat down in a college level course with math, and I was like, oh, this is awesome. This makes sense, and this is clear, and this is logical. And so yeah, PS1 was a great opportunity to get a very unique education. I took women's art history, and I went to Honduras to do service work, and I did a lot of really great things that enriched me as a human, but I didn't know that there was this whole world of math and this whole STEM world out there that I could participate in and I had a place in. It's truly a pleasure to have found it and to be here. I've done a lot of partner dancing over the years. I worked for a nonprofit back in Denver for many years, so I really love swing dancing and blues dancing, and I love a good waltz and foxtrot, but there's not a lot of young people doing that, so I always feel a little out of place when I go to those dances. I love to salsa dance. So today I'm running a Western blot, and the purpose of a Western blot is to separate out proteins of various sizes through, it's a gel that creates a mesh. And so the mesh that we're using, if you imagine this mesh kind of at the top being like a chain link fence. And as you get down towards the bottom of the gel, the mesh is like the mesh of a screen door. So as proteins move through this gel, depending on their size, they'll get stuck at different lengths. And the reason why we do this is so that we can then go through with antibodies, which are small protein molecules which can bind to these proteins, and then we can detect those with this what's called chemiluminescence, which is a chemical light-up signature that are different specific proteins. And this allows us to measure protein expression. So I've done my protein extraction, and basically what's happened is I've taken cells, tissue cultured cells, and I've put them in this buffer called RIPA buffer, which is, it's basically like a soapy buffer with some protein inhibitors in it. And what the soap does is it attacks the cell wall and causes that to kind of open up and spill the cellular guts. So we then quantify this using a colorimetric BCA assay, and I've actually saved my plate. So what we've done is I have samples of known protein concentration. So this creates kind of like a graph that will show, like, if this is this color, then that corresponds with this color, or if this color lies in between these two colors, and that'll give us an amount. So these are all my samples, and we read this with something that measures the amount of color in there. So that tells me how much of each cell lysate that I need to use for each extracted protein. So I've got my protein extracts. These have not been diluted to the appropriate concentration. I have my RIPA buffer, and then I'm not showing it, but I've gone through and I've done all of the calculations to add. So if I add this much extract and this much buffer, I want 10 nanograms to go into each well in 20 microliters. So that's kind of what's going on here. So I'm going to show you some dilutions of these samples. Make sure I get the right dilutions. And then we will load everything into our gel and run it. So for each sample, we want to make sure to give it a good mix because things settle over time. And the last thing I want to do is have spent all this time running my BCA assay and having all my proteins settle while they're sitting on ice, and then I don't load the appropriate amount. And the reason that we want to load the same amount of protein into each extract is because if there's changes in the protein level, say one cell treatment or one change in genetics causes a change in our protein of interest, we won't be able to see that if we don't have the same amount of protein loaded into each and every well. It's your job. You move clear fluids from one tube to another tube. And then somehow something magical happens. Like, oh, this kind of looks like water but smells funny. Oh, this kind of looks like water but it doesn't sit at the bottom of the tube like water. And then you mix them together and you heat them. And then stuff happens. It's like magic. It's unreal. So kind of the purpose for adding this loading buffer, this purple loading buffer, is for one, to add color to the well. Two, if you think about it, if both of your fluids are of the same weight and you pipette one into another, then fluid one is just going to kind of dissipate through. So loading buffer has something in it called glycerol, which makes it a heavier fluid. So inside the buffer that's within the Western blot gel tank, this SDS glycine buffer, which is basically like a soapy buffer with some extra salts, it'll allow it to sink to the bottom of the well. The other thing that the buffer does is, so proteins have these really unique 3D structures. And these are very carefully folded within the cell in all sorts of different places. And I'm sure some of you have heard of that, have heard of protein folding, protein processing, and if you haven't, it's no big deal. But once they're folded, protein structures can be pretty stable depending on the protein. So our loading buffer has something in it called SDS. It's also in your shampoo. When you look at your shampoo and you see lauryl sulfate, that is SDS. So it's the same stuff in my loading buffer. It's just a little cleaner and laboratory grade. We heat it with SDS, and what happens is that the protein unfolds. And it keeps it unfolded. So like so soap has a positive and a negative end, so the positive ends of the soap will stick to the negative ends of the protein and vice versa. And so that really allows us to differentiate by actual size, because you can imagine, so you've got like a really long piece of string that's like all balled up into like a million little knots versus a really straight piece of string. One's going to be bigger than the other. And when you're trying to pass it through that fence, say like the string has been starched or something, it's starched straight, that one that's all balled up with little knots is going to fall straight through, whereas the straight one isn't going to be able to go through as easily. And that can really obscure the differences in size. So it's called denaturing. So we're denaturing our proteins so that way we can really genuinely determine how big they are based upon their building blocks, their amino acid numbers, rather than, is this really tightly coiled? Does this have like a little floppy bit at the end? So this is a really important step. Syncs our proteins. It lets me see what I'm doing, which I think is probably the most important step, and it straightens everything out such that we can see it when it runs. We know that we're separating things out appropriately. Protein samples have been heated. They've been allowed to cool back to room temperature and spun down to collect any condensation. We then are going to load colored labeled proteins of known size, and this will act as a ladder. So we'll be able to see this ladder as it migrates through the gel. And ultimately what we do is, once this gel is done running, we take this gel and we use electricity to transfer all of the proteins from inside the gel onto what's essentially a fancy sheet of paper. And then that paper is then what we'll use our antibodies on, and we'll use our reporter that lights up. So for these, we just want to take it, and when you pipette, you always pipette two-handed. We just kind of stick that down into the well, and then we slowly pipette into the well. And you can see where that just kind of sits down at the bottom of the well. And then when we load this, slowly insert, and then we're going to very slowly pipette. And at the very end, there's going to be a bubble. And this bubble can blow out all of your samples. So you want to kind of bring it up and guide the bubble out. The final thing to do is to apply electricity. So our tanks have this fancy little hat, positive and negative. And what we do is we make sure that all of our stuff is aligned. So this moves back, black to black, red to red. If any of you ever have to jump a card, these are good things to know now. So we apply the hat, and then we have these power packs. And these supply our voltage. So I run all of my Westerns at 90, and that's because it creates real crisp bands. It takes a little longer, but I always feel like low and slow is the way to go. So we plug it in, again, black to black, red to red. We hit run, and we always stop and watch for a few minutes, because sometimes things throw errors, and the worst thing to do is to walk away and assume that something that's going to take 3 hours to run is running, and you come back an hour later and it hasn't run. So this will be good to go in 3 hours, and unfortunately we don't have the time to show you the rest of the protocol, but hopefully it will be great. So people used to tell me in high school that they were the best years of my life, and for me they absolutely weren't. And if I could impart one thing to young people in high school, it's that high school is not the end-all be-all, and that life spans far and wide beyond your experiences in high school. And regardless of what is occurring now, like, there will be other things to come. And it's, even if those other great things aren't immediately following, there's always an opportunity to get on the path. You know, as somebody who left high school and just kind of floundered for so many years, like, there's always that opportunity, and don't let what's going on for you now in high school be what defines your future, because it's endless. And when you're in high school, you're so young, like, you have so much time to learn and grow and experience and play and make mistakes and just not to get bogged down in high school, because there's life beyond. If I can make it, other people can make it. No one would have predicted I would be sitting here right now. So yeah, if I can do it, anyone can do it. STEM is absolutely just for everyone.